So, welcome you all to this rapid revision series, and we are dealing with seed science. So, all the important topics, whichever is possible, that we'll be discussing. It's not that we'll be discussing each and every part of the syllabus or all the PYQs. So, whichever is important that we can discuss in this 20-25 hours lectures which we are organizing. Okay. So, the another one of the important thing which is usually asked is about seed vigor and seed viability. Okay, so in that we will study the tetrazolium test. Okay, so because this is the most important test which is usually kind of done. Uh, so what happens here is that this is the chemical name 2,3,5 triphenyl tetrazolium chloride. Okay, TTZ. That's what it's called, TTZ tetrazolium test. So what happens here is that it will accept when we put the seeds in a tray, for example, and we put this TTZ. Okay, seeds are there. What will happen is the thing which you should know. So what will happen? The hydrogenase and a stable, red stable and non-sustainable substance, triphenylformazine, is formed in living cells. Like this will accept hydrogen. The living cells, whatever are there, that will accept hydrogen. Okay, will accept hydrogen, and they will give red color. Okay, they will be red in color, whichever are living, because they will accept hydrogen from the hydrogenase, and a red stable and non-diffusible substance, triphenyl formazone, is formed in the living cells. So it will stain red. But if there are some dead seeds, okay, means which are not living, so they will stain the normal. They will not be having any stain. Okay, and based on that, we can know whether the seed is viable or not. Okay, so this is the basis. So in that, the other tests, some of the other tests, please remember, not everything, one or two, so that you'll be able to write like what are the seed testing, what are the seed testing tests which are done. So in that viability and vigor, if they ask, okay, so some viability tests and some vigor tests you should know. So one is like indigo carmine test. Okay, indigo carmine test. It's usually done in trees and shrubs. So the non-living area of the embryo will be stained blue color and the living will be unstained. Understood? Second is like this you take radiographic method. Whichever is difficult, don't remember because they are not going to ask like specifically tell about this. Okay. So radiographic methods like X-rays are used. Detection of empty cell and mechanically damaged cells can be understood. Okay. By the X-ray method also we can use. It is usually used in pinus and cucurbits. And also we have another technique called excised embryo test, wherein we are using the embryo is excised from the seed, means it is removed from the seed and it is placed on a moist paper. Okay, and they are allowed to grow. It is usually done in case of a tree species, like excised embryo. Embryo is removed from it. Only in tree species, not possible in other crop species. This one, please remember, GADA. This is very important. Glutamic acid decarboxylase activity test. So what happens here it is, first rate, it is a sensitive test to measure seed deterioration in storage and to predict relative storability of seed loss. It is based on the measurement of the activity of the enzyme glutamic acid decarboxylase, which break down glutamic acid into CO2. Okay, so this glutamic acid will be broken into CO2, and what we need to do is to measurement of CO2 is an indicator of the amount of enzyme activity. Okay, based on the CO2 activity or the measurement of CO2, then we can know how much active the seeds are. So that is the basis in case of maize and wheat. I understand how it will happen, where it will happen. Don't go deep into it. Remember this much only. Okay, nobody's going to ask what is GADA test and how it is done. Remember these things. Okay. And the other important we can remember is like ATP test. It is based on luciferase activity. It is like an something like radioactivity will be there. This luciferase. Okay, based on that also we can detect which seed is having vigor and which seed is not. And also we usually use this respiration quotient. Okay, test based because only living cells will have respiration while the non-living will not have. Other test, don't worry. Okay. So next important is seed treatment. So Please know the definition. It is a treatment given to the seeds before sowing to improve the germination vigor potential along with maintaining the health of the seed. Okay, so that is the basis of seed treatment. So what are the different types? So we have some different types, like it can be chemical, okay, and it can be some insecticidal, fungicidal, okay, and also we have presently like biological also. Okay, so many different types of seed treatments are possible. So the basic first one, the chemical seed treatment. Where do we do? Okay, wherein we use different seeds with nutrient, vitamin, and micronutrients we use to test. So in case of paddy, we use 1% KCl solution for 12 hours. We keep it, okay, in something like Petri dish you take, okay, and keep this and have this 1% KCl. And in sorghum, we use 1% NaCl2. Okay, need not remember all these three, just one or two you remember, that's enough. In pulse, like we use zinc sulfate, magnesium sulfate. Okay. So next is insecticidal and fungicidal treatment. So what we do here, we use like the different chemicals which are used for fumigation and insecticide treatment. Like we can use mancozeb. Okay, mancozeb is there. Then we have Bavestin. Okay, and we can also use different insecticides that are usually uh, used for seed treatment. Okay, and also like Captan is there, Tyram is there. Okay, these all you can use. But usually we use Mancozeb and others, but Bavestin we don't use generally on seed treatment, only on the crops we use. Okay, that's correct. Next, like application of heat. That is also a way of seed treatment. Like it can be like using hot water or hot air and other things. Okay, like hot water treatment we do for loose mud. Okay, what happens is wheats are pre-soaked for 5 hours in 37 to 40 degrees Celsius. Okay, in that hot the temperature, they are pre-soaked. Okay, the seeds are pre-soaked. Okay, then they are dried. Okay, this will reduce the loose mud incidence. Also, we use hot air treatment for controlling viruses. Then solar heat treatment for like used in hot climate. Just in the outside, outside the field, you just keep the seeds. That is solar heat treatment. That's it. Okay. So next we have seed dressing. Okay, it is a treatment run for any agriculture or horticulture crops, wherein we use different methodologies. Seed dressing, like we use chemicals or disinfectants. Like what we do with chemicals is, this is what the normal procedure we do: two to 3 gram, okay, per kg of seed. We take this chemicals like maybe mancozeb or Captain or Thyran, and then we use 1 kg of seed and we mix it, okay, and they leave it for some time. This will help in seed treatment. And also we can go for seed, it will help in seed infestation, okay, seed protectant. All these are the features. What happens in case of first is this seed disinfestation, like complete eradication of fungal spore will happen in case of disinfection. But in case of disinfestation, what will happen? Destruction of surface-borne organisms that have contaminated the surface. Okay. Destruction surface-borne organisms that have contaminated seed surface, but not infected the seed, not seed surface, inside seed. Okay, which are not infected the inside part of the seed, just on the surface means disinfestation you can do. Also, some are protectants. Okay, they will have more advantage. Protectants means like till the seeds become as the seeds germinates. Okay, until the young seedlings forms. Okay, these protectants will act on them. They are not like completely like chemical fertilizers. Okay, they can be some protectants like biological ones. They are like protectants. Okay, like neem seed also. Okay, this is also important. Next, very important, special treatments. They've asked. Okay, they've asked about seed fortification. Okay, different methodologies, special treatments. These are also previous year questions. See, these are all covered in a very short time in a very crux format to help you not go anywhere and rely on many other things. Okay, here you see the first is seed hardening treatment. Okay, what is hardening? So seeds are exposed to tolerate like initial drought and cold. Once seeds are exposed to these things, then it is believed that in the later phase of life, it will germinate well and it will have good yield and other features. Okay. So, so this is the ratio which is that is not required. Majorly just focus on this. Seed mainly to tolerate initial cold, drought or heat that will be able to survive better. Next is seed fortification. This is very, very important. What happens, it supplies nutrients to seed, thereby achieving the vigor. Means the seed on the seed outer layer, we will have some sort of nutrients. Okay, we will have some sort of nutrients, some sort of uh, polyproteins and all. For example, we use here magnesium sulfate. Okay, which is like an oxidation and reduction potential of the cell. Okay, it will increase the oxidation potential of the cell, and this will lead to increased germination, better disease resistance, all these things. Fortification means increasing nutrient content. Remember that. Next is moist sand conditioning. Okay, here what we do, it is like need based and concentration chemical 2 to 4 solution. Here we use proteinaceous seeds are mixed with moist sand. So the seeds which are proteinaceous, like the seeds, okay, then they are mixed with sand, okay, and then they are sown. So this is called moist sand conditioning. Next is also very, very important. What is it? Seed pelleting. Pellet means what? Pellet means where we do these things, some seed like mustard, very, very, very small. Okay. So while sowing also, it is not properly sown. There may be some damage to the seed. So what we do, so this is seed. Okay. This, for example, take this seed. Okay. Very small. So what we do, so here there will be some coating we will do. Okay. This coating. So this will be a nutrient coating, okay, on the seed. So what will happen? It will handling will improve. You can easily sow this seed. It will be like very, very, then it will be different. It will not be sticky. Then you can easily sow. And also it will, what it will do, it is nutrient mixture is there. It will increase seed size and the seed rate also. It is mainly done in small seeds. Okay. Next is like seed infusion. Okay. What we do here is infusing, seed infusing. Okay. So what we do, organic solvents like acetone and dichloromethane is used to infusion of nutrient growth protecting substance into the seeds. Here the treatment of organic solvent slowly gets evaporated, hence seed drying is not needed. Commonly used to break dormancy. Okay, this technique is usually used to break dormancy, and the chemicals which we use, you should remember, is acetone and dichloromethane. Okay, and it will use in the infusion of nutrients and growth substances into the seed. So because of these chemicals, it will help the nutrient movement. Okay, and then this one last you remember. These both are not like very much important. This is important. Osmotic priming. So this is usually done in some legumes like peas and bean, and that have high protein content and large imri. Okay, here uh, this treatment why it is made, so to imbibe water. This is done to imbibe water because the seed coat would be hard and it will not be able to go and to germinate. So we use this chemical polyethylene glycol, which is very important chemical. Polyethylene glycol, even you will study in dormancy chapter also. It is used for breaking dormancy. Okay, so when we use this chemical, then the imbibition of the water will increase, by which there will be better germination. Okay, and also this osmotic priming will lead to uniform and early field emergence. Okay. So deep dormancy, I have already made one video. And the next important is cultivar identification. Okay. So you would have seen about the genetic principles. Okay. And genetic methodologies. So these are also the basis on how the cultivars are identified. On the field, how it is identified. Okay, and also how do you differentiate cultivars from one another? Maybe you may use some molecular tools, you may use some biochemical tools, you may use some morphological tools, you may use some field tools. All these things we will see. Okay. So what is cultivar? Cultivar refers to an assemblage of plants selected for desirable characters that are maintained during propagation. Okay, they're just varieties, or cultivars means just anything which is like seed material that is available. Okay, then the identification of cultivar is based on physical, physiological, biochemical, and many other features which we will discuss. The first thing examination seed in laboratory, morphological character. How you differentiate a seed? That is the basis. Okay. What happens here is morphological character, like maybe color. Based on color, you can differentiate. Second, it can be based on some shape. Okay. It can be based on seed size. It can be based on texture. Texture means how you touch and feel that rough surface will be there or not. Based on that, we can differentiate a variety. That is what it is telling. Nothing big thing. Okay. Another is physiological test. Physiological means the physiology, like the respiration, photosynthesis, how they are affecting it. So we go for something called fluorescent test. Okay. In the dark room, we go for fluorescent test, and based on the illumination, we can identify them. Okay. And also we can go for disease resistance test based on the susceptibility of the disease after inoculation. Inoculation means you put the disease intentionally and check. Okay. And based on that also you can see which variety is susceptible, which variety is resistant. And also we go for biochemical test like phenol color reaction test. Okay. We use different chemicals. Like you should remember this chemical phenol. Okay. Which is used to identify, distinguish wheat and paddy. Okay. Don't remember the procedures. Just remember what are the, because they will ask, if even if they ask, they will ask in very broad question. No need to remember this 18 hours you have to place. No, no, no. Don't remember. Just where it is used in wheat and rice based on color intensity developed on the seed coat, because we will treat with this chemical. Okay, phenol solution 2% for 16 to 18 hours. We'll be using it, and then we'll be using it on the filter paper. So what will happen? Based on the color intensity, we'll be able to differentiate the cultivars. Okay, based on many chemical procedures. There is no need. Okay, how they are done, other techniques. These are some examples only, like peroxidase test we use for soybean. Electrophoresis. This is like important, like how in gel electrophoresis. Okay, how you are differentiating varieties. That is one. Next, you can also do it in greenhouse chamber. Okay, and this is very important. Field plot test or grow out test. So what we do, a genetically superior variety, we grow in the field. Okay, and based on the field observations, like it may be some morphological observation. Okay, so means our variety, I have developed a variety A. So it will have the awns will be in this some different shapes. Okay, or I have developed in this variety, it is disease resistant to some downy mildew. Then I will check for that. So like that, we are able to differentiate a cultivar. That is the basis on grow out test. So genetic purity also we will know by grow out test. Okay, in the field we will sow and we will know. Okay, based on that we will distinguish characters, and the grow out test is used to determine that. Okay, so you have done what we have done. We have done morphological test, physiological, biochemical. Okay. Biochemical, electrophoresis, peroxidase test. You just remember for soybean it is done. Means we use different everything is similar. Just, or you can just by heart the name, like sodium hydroxide test where we use. Okay, usually I think this we use in wheat. Okay, this we use in soybean. This electrophoresis is a molecular tool, not a molecular means we use molecular techniques and then we run it on a gel electrophoresis based on the banding pattern. Okay, we are able to differentiate. Next, we will see some of the seed vigor tests. Okay, first we'll start from seedling vigor test. What is seedling vigor? It is defined as the sum total of those properties of the seed which determine the potential level of performance and activity of non-dormant seed. Okay, seed cloud during germination and seedling emergence. It is the vigor. Okay. It is the speedness in which the seed is germinating. Okay. What happens in viability? Okay, viability means it is living, that's it. But in vigor, it is the speed in which the plant is getting established, how it is germinating. Okay, which determine the potential level of performance and the activity of an seed. Okay, that is done by seedling vigor test. So here also simple things like physical test we do, like based on the physical things, like we see the seed, whether it is a normal seed or not. Next, physical soundness, like whether there is any abnormalities, whether there is some tests we will do. Okay, these all things also mainly we will do like in case of while submitting our seeds. Okay, for certification, they will all check these things, whether the seed is really worth or not for release. Then we can also do for X-ray test, like we do for humans X-ray and check. So in that way, we will do X-ray and see whether the seed is having any damage or some pest attack or not. Okay, then we have some performance tests. This is very simple, like personal performance with, like as someone is running a race. So we will do some performance tests like that. First count test. Okay, based on the germination percentage on the first day. Whoever germinates first, they are better. That's what. Okay, and speed of germination. Who is germinating first in the speed within 24 hours? Okay, and also based on growth rate and seedling dry weight. So there are some 10 lines. Okay, so we will check, okay, this line is best, this line generated first, this is first. So based on that we will say, okay, this is having good vigor. That is a simple concept as that. And also this is stress test is there. What is stress test? Stress test means see, like you just imagine that you are going for some training. Okay, some police academy training. Okay, then they will do all these tests. They will see our physical, they will do medical na, when you go for UPSC you will have medical, so something like that. Second, they will also see our strength, weakness. Okay, and then stress test. Stress test means like for example some of the IFS and all forest, you need to stay in the forest for few days. Okay, such things also will be there. With the limited food you have to survive, in even in IPS, all these things. So like that, it is like cold test. Okay, we are giving cold stress to the seeds and checking the performance, whether it is able to germinate. Brick gravel test. Here what we do, the crushed brick gravels, crushed brick gravels, we will crush them and then put on the seeds. Okay, seeds are there, so on that we will put all these bricks. So we will check which is germinating well. Okay, the best ones, that's it. Okay. Then paper piercing test. Okay, we will have a paper. Okay, down the seeds will be there, so whether which is able to germinate better. Okay, so these are the basic things which are there. Then we also have accelerated aging test. Okay, so in this test, what happens is the based on the like these, like under these extreme conditions. Okay, we try to germinate them, and the rate of seed deterioration is used to know which seed is deteriorating first. Okay, because what happens is here we are exposing them to high temperature and high RH because it is not good for seeds. High temperature and high RH is not good for seeds, but you are exposing them by which it will be aging, it will start to deteriorate. So based on that, we will see which is deteriorating first. Out of the 5 seeds, which is deteriorating first, based on that, we will